All right, welcome back. Today we're pulling off a high-stakes mission, a molecular heist, if you will. Our target? The actual building blocks of life. Our goal is to sneak into a bacterial culture and get out with pure plasmid DNA. Let's get into it.
So that's really the core problem, isn't it? The treasure we're after, this plasmid DNA, is unbelievably valuable, but it's locked away inside billions, and I mean billions, of tiny little vaults, the bacterial cells themselves. To get it out, you need precision, you need the right tools, and yeah, you need a little bit of chemical brute force.
So what exactly is this treasure we're talking about? Well, a plasmid is this little circular loop of DNA. You can think of it like a bonus cheat sheet or a portable hard drive that bacteria carry around. For scientists, it's an absolute treasure trove because we can use it to carry specific genes, making it one of the most fundamental tools in all of genetic engineering. Okay, so every good heist starts with a plan. And part one is always the same. You got to case the joint. For us, that means rounding up all of our tiny little vaults. We have to gather billions of these bacteria cells from the liquid they're floating in and get them all concentrated in one place. This is the roundup.
And here's how we do it. We take our sample and we stick it in a centrifuge, a machine that spins it at an insane 12,000 revolutions per minute. This crazy force shoves all the heavier bacterial cells down to the very bottom of the tube, where they form this dense little clump we call a pellet. Then you can just pour off the liquid and boom, all our vaults are gathered together.
All right, now for the really fun part. We've got our vaults collected, so it's time for the break-in. Now, we can't just smash these things open, right? We need a really sophisticated set of chemical tools to basically dissolve the cell walls without accidentally shredding the treasure that's inside.
And here is our toolkit. It's just 3 solutions, but each one has a very specific job. You can think of solution I as our prep guy, getting the cells ready. Solution 2, that's our dynamite. It blasts the cells wide open. And solution 3 is our cleanup crew, coming in right after to neutralize the chaos and separate the good stuff from all the wreckage.
First up, we add solution I. Its whole purpose is to get that tightly packed pellet of cells off the bottom of the tube and get them all floating around freely in the liquid again. We have to mix it up until every last cell is back in suspension, totally prepped for the next phase of the operation. Now, this is one of those little details that sound small, but it is absolutely critical. Our source material makes a specific note about this. Resuspend thoroughly such that no clumps remain. And there's a good reason for that. If any of our vaults are still stuck together in a clump, our chemical dynamite just won't be able to get to them, and all the treasure inside will be totally lost.
And now for the main event. We add solution 2. This stuff is a really powerful detergent that literally rips the cell walls and membranes to shreds. It's a process called lysis. We have to mix it gently though, just by turning the tube upside down a few times, because if we're too rough, we could accidentally tear up our fragile plasmid DNA right along with all the other junk.
Okay, bang, the cells are open, but now we've got this chaotic soup of DNA, cell walls, proteins, just everything all mixed together. So we immediately add solution 3. This stuff is an acidic solution that neutralizes that explosive reaction from solution 2. This causes all the big, heavy chromosomal DNA and the other cell debris to just clump together into this gross, messy white glob. But, and this is the clever part, our tiny, tough little plasmids stay perfectly dissolved in the liquid. And this next spin is where the separation really happens. All that unwanted junk gets packed down into a hard, solid pellet at the bottom. The clear liquid that's left on top, we call that the supernatant. That's our liquid gold. That's where the plasmids are. We just have to carefully suck that liquid up with a pipette and move it to a clean tube, leaving all that wreckage behind. So we've successfully separated the treasure from the trash, but we have a new problem. It's completely invisible, just dissolved in this clear liquid. So the getaway phase of our heist is all about turning that invisible prize into something solid that we can actually see and, collect. Honestly, this is my favorite part of the entire process. It always feels a little bit like a magic trick. We're about to force our DNA treasure to literally crash out of the solution and finally reveal itself. So how do we pull this off? Simple. We add alcohol. See, DNA is not soluble in alcohol. It hates it. So the second you add it, the DNA immediately precipitates. It turns into a solid. After a good mix, we do one last super high-speed spin for a full 10 minutes. This forces all of that newly solid DNA down into a tiny, almost invisible white pellet at the very, very bottom of the tube. The heist is almost complete. We've got the treasure in hand, but it's not quite ready yet.
It's kind of like finding a diamond that's still covered in mud. So this final phase, the cleanup, is where we wash our DNA to make sure it is absolutely pure and ready for its next big assignment.
You can think of this step as basically washing off the fingerprints. We pour off the isopropanol, and then we add a little bit of 70% ethanol. This washes away any leftover salts or other chemical impurities that might have hitched a ride. We give it one more quick spin to pack the pellet down, and then pour off that ethanol wash.
Now, this part is simple but important. We just let the pellet air dry completely. We have to get rid of every last trace of that ethanol. Once it's totally dry, we dissolve our pure, clean DNA in a special liquid called TE buffer. This buffer will protect it and keep it stable for long-term storage.
And I have to point out this final warning. Again, straight from our source material. It's a big one. If you don't let that pellet dry completely, any leftover ethanol can seriously mess up future experiments. It basically poisons the enzymes you might want to use later. It could ruin everything you plan to do with that DNA, making the entire heist a complete failure. Every single detail matters.
So, we've pulled it off. We went from a cloudy tube of bacteria to a tiny, pure pellet of our treasure, now stored safely in a buffer. But why? Why go through all this meticulous trouble? What's the big payoff?
Well, the payoff is huge. This procedure, which seems kind of simple, is actually the starting point for some of the most important science happening in the world today. That tiny pellet of DNA is the key that unlocks genetic engineering. It lets us create life-saving medicines, like insulin. It helps us understand and fight diseases. It fuels countless new biotechnologies. It all starts right here, with this process.
And really, that's the ultimate takeaway. This isn't just some boring lab procedure. It's the recovery of a blueprint for life itself. The heist is over, the code has been cracked, and the treasure is finally ours. So the only question that's left is, what incredible things are we going to build with it?